So looking at the, all right, we're going live.
Alrighty.
All right, good afternoon and welcome back to session 2 of Cosyne 2021. Undoubtedly a success already, with 1,200 viewers in session 1 and already 1,100 live right now. Pretty much double the audience of your regular Cosyne event. Maybe actually online conferences and seminar series are pretty much the only thing positive coming out of this pandemic that is good. So hello to all of you in various stages of lockdown. Glad I don't have to see you all. Socializing is for suckers anyway. So apologies to those of you who have to look at me. It's going to change right now. I'm going to introduce your first speaker, Colin Bredenberg from Eero Simoncelli and Cristina Savin's lab of New York University. Colin is a graduate student and he's going to talk about impression learning, online predictive coding with synaptic plasticity. Colin, the floor is yours.
All right, thank you. Let me bring up my presentation. We good? All right. So today I'm going to be talking about learning Bayesian sensory representations using synaptic plasticity. In this paradigm, partially observable objects in the environment, like this cube, produce sensory stimuli. So here, a green square on a white background. Given the stimulus, we might imagine that the brain performs a computation to infer a full cube from the observed two-dimensional retinal stimuli. We can formalize this process using probability theory, where the hidden variables are present in the environment with some probability distribution P of Z, and we have a conditional probability distribution P of S given Z, describing how those hidden variables map to sensory stimuli. We might imagine that the brain can exploit the statistics of its inputs, so that when it's presented with a stimulus S, it infers a probability distribution P of Z given S using Bayes' theorem. However, this has two problems. First, the brain doesn't have access to the true probability distributions of hidden variables or stimuli in the environment. At best, it is an approximation. And second, actually computing a posterior distribution according to Bayes' theorem is in general very difficult because it involves integrating over a potentially very high-dimensional stimulus space. So to fix this situation, we'll revise our original diagram by assuming the brain builds its own model of hidden variables in the environment called PM of R. And it maps these hidden variables to stimuli that it receives through PM of S given R. Then, when the network receives a stimulus, it performs inference with respect to its own model, not the true model of the stimuli. And we're denoting the inference distribution by Q of R given S to make clear that the network is performing approximate inference, not explicit inference, because, again, explicitly performing Bayesian inference is in general intractable. Given this setup, learning amounts to matching the brain's model to data and its approximate inference to the correct inference. In this talk, I'm going to present a potential mechanism for how the brain could use synaptic plasticity to perform this learning. So, to be more concrete, we'll translate these two requirements into the language of optimization. We'll assume that the network is attempting to minimize a loss L, which has two components. The first minimizes the KL divergence from the approximate model to the true data distribution, and the second minimizes the KL divergence from the approximate inference distribution Q to the correct inference procedure for PM. For those of you that aren't familiar with the KL divergence, you can think of it roughly as a distance metric between two probability distributions. We're trying to bring these two sets of probability distributions close to one another. So now that we've recast our goal as optimizing an objective function, we can focus on the biological implementation. What do P, M, and Q correspond to in the brain, and can we map learning onto biology? So we're going to use model pyramidal neurons with apical and basal dendrites with distinct functional roles. The apical dendrites receive top-down and recurrent inputs and are dedicated to learning a model of the data, while the basal dendrites receive feed-forward inputs and are used for inference. We're also going to include an inhibitory gate on each neuron, which we're going to call delta, which controls whether incoming current from apical or basal synapses drives the dynamics of the neuron. And for those who are curious, there is some experimental evidence for each of these three components. We can formalize the dynamics of our example neuron with the following equation, where V apical is the voltage in our apical compartment, V basal is the voltage in our basal compartment. The activity is determined by one of these two signals depending on delta, with noise added on top. Now, because there's noise added on top, neural activities are samples from a probability distribution. And in fact, when delta equals 1, we're sampling from Q, and when delta equals 0, we're sampling from PM. That's our model. We're also going to include synaptic plasticity at both the apical and basal dendrites, which are going to be very similar to one another. Plasticity at the apical dendrites has post and presynaptic terms, and this would be roughly Hebbian, except that the local dendritic inputs are subtracted out from the postsynaptic firing rate of the neuron. Plasticity at the basal synapses is identical, except that the dendritic inputs subtracted out are due to V basal instead of V apical. So that means that the plasticity rules have an identical form, but depending on the inputs received locally to the dendritic compartment and the gating state of delta, their behavior can be very different. So our architecture is such that individual units all receive feed-forward inputs from a sensory layer and send back a prediction signal attempting to match that signal. Additionally, the presence of gating by delta means that our neurons can be in two different modes. In the first mode, delta equals 1, activity is dominated by feed-forward stimuli projecting to basal synapses, and neural firing rates correspond to noisy samples from Q of R given S, which is our approximate inference distribution. In the second mode, delta equals 0, activity is dominated by recurrent and top-down connections projecting to the apical synapses, and neural firing rates correspond to samples from PM of R. That means the activity is sampling from the model's prior distribution. So previous work on the wake-sleep algorithm has shown that these two different modes of activity can be used to minimize the loss that I showed on the previous slide through local synaptic plasticity. But the trouble with the wake-sleep algorithm is its sleep phase. It requires lengthy dreamlike experiences for the basal synapses to learn, and an animal could not possibly be actively perceiving during this time. So our question in this study has been, can these phases be folded together so that animals can actively perceive and learn to build representations of their stimuli concurrently? And if they can be folded together, how much would it disrupt perception? So our idea is to have very brief pulses of apical activity interleaved with active feed-forward input, so that the network's activity is always tied to its current impression of the sensory inputs. And for this reason, we've dubbed our learning algorithm impression learning. So now that we've described our learning algorithm, it's time to analyze its capabilities. We're going to construct a set of artificial stimuli with parameters that we can control so we can see how learning occurs under a variety of circumstances. The hidden variables that generate our stimuli are uncorrelated Ornstein-Uhlenbeck processes. That means that they're temporally varying stimuli, and I'm showing one right here, whose steady state distributions are Gaussian. And we can map these uncorrelated hidden variables into high dimensional stimuli. So here we have 10 hidden variables and 40 observed variables, each with its own row with the color indicating the magnitude of the signal. And these observed variables become potentially strongly correlated with one another. So the stimuli are fed into our network by feed-forward connections, and the network attempts to uncover the original uncorrelated hidden variables. To verify that our network is properly learning to represent the hidden variables for these stimuli, we need to look at a few things. First, we can look at our loss, and we can see that it's decreasing over learning, which is a good sign, but the network could still be in a poorly performing local minimum. Looking a little deeper, if the network is accurately building a sensory representation, it should function as a good autoencoder, meaning that its learned hidden states should map back accurately to the observed stimuli as they're presented, when the network is in inference mode. That should mean that an example stimulus and its reconstruction should overlap near perfectly, which we see to be the case, both for a particular stimulus dimension that I'm showing here, and across many different stimulus dimensions. So in this plot, stimuli and reconstructions should lie along the identity line if the learned representation is good. Lastly, if neural activities are representative of hidden variables in the environment, the autocorrelations of neurons should correspond to the autocorrelations of hidden variables that produced our stimuli. So this should be true in both the inference mode and in the modeling mode, because if the prior has been learned correctly, it should reflect the true statistics of the data. So in this last figure, we see that this is the case, where both inferred autocorrelations and model-simulated autocorrelations closely overlap with the true hidden variables. So having validated that learning works for a simple stimuli, we can now compare it to classical methods. So we'll be looking at two learning algorithms in particular, reinforce which is a biologically plausible learning algorithm that combines a global reward signal with local Hebbian coactivity, and backpropagation, which is the standard but biologically implausible method for training synaptic weights in artificial neural networks. Both provide unbiased gradient estimates, but reinforce requires more data samples and much lower learning rates to improve performance, especially in high dimensions because of its high variance. Because reinforce is biologically plausible and backprop is not, we should think of reinforce as a more realistic baseline and backprop as a gold standard. Our algorithm, unlike these two methods, is biased, so it doesn't necessarily point in the true gradient of the loss. We need to validate experimentally that it aligns to proper gradient descent, which we're going to assess using Cosyne similarity, and that it has, so a positive Cosyne similarity, and that it has variance properties that are closer to backpropagation than reinforce, which we're going to assess using the signal-to-noise ratio of our gradient estimates. Also, we need to assess that it learns effectively in high-dimensional environments. So we have exactly those necessary validations here. I'm plotting first the loss through time as the network learns its task using impression learning. And underneath, I'm showing the cosine similarity between the impression learning gradient and the backpropagation gradient. We can see that the network begins with a positive alignment, increases its alignment throughout time, before losing some of its similarity as performance becomes near optimal, where the norm of the gradient is already near zero. Underneath, I'm plotting the signal-to-noise ratio averaged across synapses for impression learning, backpropagation, and reinforce. And notice that reinforce has a much smaller signal-to-noise ratio, but keep in mind that signal-to-noise ratio is only an indirect measure of performance. So in this last plot, we're going to validate that reinforce performs much worse as we increase the dimensionality of the hidden variables in the stimulus, and that's relative to impression learning, which is demonstrating again our algorithm's superior efficiency. So now that we've shown that our algorithm is effective, we can address our second question. How much does our online learning algorithm affect ordinary perception? So for the algorithm that we've been using up to this point, where alternation is rapid on a time-step to time-step basis, we can plot a network trace when it's fully in inference mode, delta equals 1, and compare that to alternating modes in blue. And you can see that the neural activities show the same general trends with minor deviations in activity. Now, the longer our network is allowed to stay in simulation mode, the more it's going to disrupt perception. So if we instead switch modes every 31 time steps instead of 1, you can see that the neural traces deviate much more significantly from the proper inference mode. That means that the approximation depends on how long you stay in each mode, which we're going to summarize in this figure. So for the blue trace, impression learning neural activations have a very strong correlation with the inference mode shown here. But the longer the switch period, the worse the correlation. And we can also see that the loss throughout time in our two extreme cases are very close to one another with similar asymptotic performance. And that's important because the wake-sleep learning algorithm is effectively equivalent to setting a long switch period in our method. That means we can learn almost as effectively as the wake-sleep algorithm, but can do it continuously with perception instead of having to resort to a distinct sleep phase. So it's very important to assess these types of learning algorithms on realistic data sets for the sensory modality in question, because performance can vary radically as the stimulus complexity changes. So we're starting this process by scaling our algorithm to processing static MNIST images for our same one-layer network architecture. And as you can see, performance is again much better than reinforce. Additionally, for the static MNIST, we can see that our network is able to produce very good reconstructions of the stimuli that it receives, showing that our learning algorithm is able to scale to higher dimensions and more complicated stimuli. So having demonstrated the efficacy of our network for the simplified architecture, our hope is to scale our network to the point that it can produce similarly convincing results for natural videos. So having explored the properties of our algorithm, let's focus for a moment on its relationship to biology and previous theoretical work. Our model posits that the apical dendrites receive recurrent and top-down signals, whereas basal dendrites receive feed-forward sensory signals. And this has been consistent with experimental work, and has also been suggested by previous work trying to provide an account of supervised learning in the brain. Also, there's some evidence that apical stimulation of pyramidal neurons induces synaptic plasticity at basal synapses. And these features are also compatible with our model, as well as other approaches to unsupervised Bayesian learning. Now, unlike wake-sleep learning and many other algorithms, our model allows for learning continuously with perception. From our model's construction, we can make two distinguishing predictions. First, there needs to be a neural mechanism for rapid control of the apical or basal influence on activity, that's delta. And second, increasing the relative influence of apical synapses should induce structured hallucinations, meaning hallucinations that are believable percepts tied to reality, which should have not tied to reality, which should have consequences both perceptually and at a neural population level. So to close off, I would like to thank everyone in both of my labs, that's Eero Simoncelli's lab, as well as Cristina Savin's lab, for providing a wonderful working environment and supporting me in 100 ways. Thank you all for your time.
Thank you, Colin. Thank you for a lovely talk. The audience, maybe you should know, we have a 15-second delay before you hear us, so your questions are going to take a little while if you're typing them now. In the meantime, I wanted you to clarify for me the changes in delta that you're making to switch between the two modes. Are those 0 and 1, or are they gradual changes?
Yes. So in the setup that we currently have, and to maybe mute your mic, thanks. In the setup that we currently have, deltas are hard 0s and 1s, and it's actually something I'm really interested in exploring, whether or not you can have partial, like 0.5 or something like that. And I think that we have some preliminary results indicating that it's possible, but as of now, we've just been sticking to strict 0s and 1s just to validate that the learning algorithm works. Yeah.
All right. We have questions coming in now. We have a little bit of time. Is there evidence for overfitting in your procedure?
So overfitting, not really, because we're working in particular with synthetic stimuli. We're working with OU processes that we're generating, and so when we're showing the networks these stimuli during training, they'll only ever see a particular stimulus once, and there's no possibility of overfitting to a limited data set. The data set's effectively infinite because we're synthesizing our data. Yeah.
Next question by Linxing Preston Jiang. What's the motivation of using the switch period instead of a more feed-forward processing like a VAE, so inference and learning can happen at the same time? Sorry, the window switched while I was reading.
Right. So the difficulty is that when you try and formulate this evidence lower bound objective function that we wrote out in the first few slides, if you do gradient descent on that, just in the most simplistic way that you could imagine, if you do gradient descent like you would for a variational autoencoder, you're going to end up being forced to do something non-local. You're going to end up with either a global reward signal, like in reinforce, or you're going to have to do backpropagation. Algorithms that try and produce a local synaptic plasticity rule typically have to use some kind of cheat or shortcut in order to produce a local synaptic weight update. And normally the route that's been done is through introducing these extra phases of learning. And so it would be good if you could learn completely continuously and not have these alternating phases. But as we're doing right now, we're improving upon basically the existing state of the art in synaptic plasticity. Yeah.
Thank you. Jason Moore has a question that got upvoted quite a bit. What biological mechanism do you have in mind for gating between the two states?
Yeah, so a little bit. This Matthew Larkham's 2013 paper suggests that a potential mechanism for this kind of gating could be inhibitory neurons that specifically target the apical dendrites. We're currently postulating that delta needs to be a global signal that is, it's affecting all neurons in the network equally. And we're also working on relaxing that assumption so that individual neurons might be in a different state from their surrounding neurons.
Thank you, Colin. Thank you. I'm afraid we're out of time.
Of course.
The questions will be available for you to answer maybe later. But for now, thank you again for your talk. Lovely. And we're asking Sebi Rolotti to come online
Thank you.
now and switch on his camera. Good afternoon. Sebi Rolotti is going to talk about inhibitory feedback control of behavioral timescale, synaptic plasticity induction in CA1. Sebi is currently at Columbia University in Attila Losonczy's lab. Sorry, I butchered that. Go ahead.
I think he's very used to that. Thank you. Okay, so yeah, thanks so much to the organizers and to everyone attending and participating. I'm Sebi Rolotti. Today I'm going to talk to you about ongoing work in the Losonczy lab, looking into local circuit modulation of place field formation.
Sebi, sorry to interrupt you. We're seeing your share screen. We're not seeing your presentation screen.
Alas. Oh no. I feel like we tried this multiple times. No. All right, let's try again. Is that better?
Lovely, lovely. That's perfect.
All right. Many apologies. Okay. I'll just jump right into it. All right, so it's hopefully no surprise to anyone in the audience that pyramidal cells in area CA1 of the hippocampus tend to be tuned to space. It's about a third of the cells in any given environment. The place cells, firing specifically when, in this case, a mouse, runs through that cell's preferred location, or the place field. So until recently, however, not much has been known about how these representations form in the first place. And important advances on this problem came pretty recently with an observation from Katie Bittner of the McGee lab while doing intracellular recordings of CA1 pyramidal cells as a mouse ran down a linear track. So she found that previously silent cells would sometimes display large plateau potentials, likely a somatic signature of a multi-branch spike in the apical tuft, and then subsequently form a place field, evidenced by reliable burst firing at that location on later laps. So importantly, they found that repeatedly inducing these plateaus at a fixed location via current injection could also cause the formation of a place field at that location. Follow-up work identified this phenomenon as requiring a novel form of plasticity, behavioral timescale synaptic plasticity. So BTSP has a number of really interesting, unique features. So first, it's a small number of associative pairings needed. So it was 1 to 5 in the examples I just showed you on the last slide. And then furthermore, the plasticity kernel they identified is asymmetric. So it's a longer tail for inputs prior to the plateau spike, and the kernel is seconds long. So these two features in turn predict that, 1, because of asymmetry, induced fields should peak prior to the location of the plateau. And then 2, because of the seconds over which inputs can be potentiated, the width of the induced field should be correlated to the speed of the animal during the plateau spike. So all this is corroborated by the data. And so this suggests a unique and powerful form of plasticity that's primarily responsible for place field formation and that is capable of generating new representation for any location in the environment. More recent work, though, from the Bujaki lab presents a slightly contradictory picture. So in similar experiments in which now a slightly larger number of cells are repeatedly stimulated at a fixed location via a micro-LED with a approximately 125 micron stimulation radius, researchers found that stimulated cells did not preferentially form place fields at the stimulation location. And instead, a large subset of both stimulated and unstimulated cells underwent remapping. And the location of the post-remapping place field was predicted by weak firing prior to stimulation. So while it's not clear why differences in the experimental protocol here, weaker stimulation optically delivered to a larger number of cells, could not produce place fields at arbitrary locations, and instead they were constrained by prior network activity. So it's unclear what mechanisms the circuit is bringing to bear in organizing its representations to balance the need on the one hand for arbitrary representations against, on the other, the inertia of a bias for existing representations. We already know, and it's already appreciated, that the circuit is somehow able to scale the rate of place field formation along this continuum in a context and behavior-dependent manner. So we know that, for example, place fields initially form more rapidly in novel environments, and this process slows with familiarity. And we also know from work from our own lab and a number of other studies that in experiments in which the mouse must learn the location of a reward, place cells preferentially form near new reward locations. So this leads us to a number of important questions. How can we explain these contradictory results regarding experimentally induced place fields? More generally, what are the constraints on arbitrary place field formation, and what is the mechanism by which the CA1 circuit is capable of scaling the rate of place field formation across the population? And then finally, what can all this tell us about the relationship between place field formation and learning and behavior? So to first determine if we could optically induce place fields at all, we tried to replicate the single-cell Bittner experiments as closely as possible. So to this end, we turned to a technique, single-cell electroporation, which allowed us to specifically deliver plasmids to small groups of cells and thereby express an opsin, here carmine, and a calcium sensor, GCAM7B, in exactly 1 or sometimes 2 pyramidal cells in CA1. The experimental protocol we use is super similar to those I described in the Bittner and McKenzie studies. So a head-fixed mouse is running on an approximately 2-meter Q-rich belt for random water rewards. And after 10 baseline pre-laps, the sparsely labeled cells are going to be repeatedly stimulated at a fixed location by 1 second of wide-field LED light delivery for 5 laps in a row. So after this stim epoch, the stimulated cells are then monitored for the remainder of the 20-minute session, which we'll call post. So we can look at an example induction session here with a single cell. And here's a few seconds of Z-scored fluorescence from that cell at what will be the eventual stimulation zone on laps 3, 6, and 9. The cell is, hopefully you can see, is pretty silent at this location. Then during stim, we see the cell strongly firing in response to light. And already by the 3rd lap of stimulation, here lap 12, we can see ramping firing preceding light delivery. On the remaining laps, a place field has developed near the stimulation zone, and the cell continues to reliably fire here, even during a second exposure at the belt 24 hours later. So we can measure the effect of stimulation on a cell's tuning in a number of different ways. So here I'll quickly introduce the 3 metrics we'll use for the rest of the study. Here we have a cartoon of a cell's tuning curves during pre and then immediately after stimulation during post. So first we can ask during each of these epochs how far the center of mass of the activity, or the activity centroid, is from the stimulation zone. We can also ask about the delta of these distances, that is, how much did the activity centroid shift towards the stimulation zone as a result of stimulation. And then finally, we can ask specifically about the formation of a classifiable place field near the stimulation zone. So that is, does the activity during post meet reliability and spatial information criteria in order for it to be classified as a place field? And if so, is that place field located within 1/10 of the belt length of the stimulation zone? So according to all of these metrics, this optical stimulation protocol clearly biased activity of stimulated cells towards the stimulation zone. So we can see that activity that was no closer than chance during the pre is significantly closer during post. And this can also be seen in the shift, which is positive for 8 out of 9 cells. And then finally, by computing place fields on all these cells, we find that 7 out of 9 of the stimulated cells had new fields near the stimulation zone during post, and 2 of those cells still had fields there up to 72 hours later. Importantly, these new fields had all the features of BTSP I previously described. So they formed after at most 5 pairings of place and stimulation. They all had peaks just prior to the stimulation zone. And the place field widths are strongly correlated with the velocity of the mouse during the seconds around that first stimulation. So these results from single cells, along with other data I don't have time to present, which we found no overrepresentation of the stimulation zone when all pyramidal cells in the field of view were stimulated, suggest that stimulation sparsity may somehow play an important role in governing the efficacy of BTSP induction. So we hypothesized that this might be to increase recruitment of local dendrite targeting inhibition, preventing the generation of dendritic spikes at higher densities, and therefore the induction of BTSP. So to test this, we turned to an approach to more carefully titrate cell stimulation sparsity by titrating the sparsity of opsin expression. So for this, we used a viral approach in which a tamoxifen-inducible CRE is targeted to pyramidal cells using a KMK2 promoter. And alongside that, we're going to inject a CRE-dependent carmine and a CRE-independent GCAMP that's going to allow us to monitor both stimulated and unstimulated cells. So this is what that expression looks like. Note the broad GCAMP expression and the significantly sparser opsin co-expression here in about 5% of the pyramidals. And so first, to look for increased recruitment of interneurons at higher pyramidal cell stimulation densities, we're going to do a super simple experiment in which we deliver 1 second of wide field stimulation at random times while imaging either putative interneurons in stratum oriens or pyramidal cells in stratum pyramidal. And then by slowly increasing the tamoxifen dose every few days, we can increase the density of pyramidal cells responding to light, and then look for a concomitant increase in the interneuron activity. And that's exactly what we find. So here on the left, we have the mean PSTH for all interneurons in an example mouse across 5 sessions with increasing stimulation densities, with a very large and prominent change in responsiveness from session 1 in black to session 5 in the lightest gray. And on the right, across all sessions, or colored by mouse, we found a very strong correlation between the interneuron response size and the fraction of pyramidal cells that were responsive to light stimulation in the field of view. So these data together argue that stimulation density-dependent increases in recurrent inhibition are at least a plausible mechanism for gating BTSP across the population. So we therefore turn to the same viral strategy to look at BTSP induction efficacy in subpopulations of cells at varying stimulation densities. So further supporting our model, we found a really strong relationship between the bias of stimulated cells towards the stim zone and the fraction of pyramidal cells being stimulated during the induction protocol. That's on the x-axis. So here each point is the mean across all stimulated cells in the field of view. And so to see this even more clearly, we can separate these experiments according to a median split on the stimulation density. So while stimulated cells in the medium density experiments, so those with about 3% density or higher, looked no different from chance or from the unstimulated cells, which I'm not showing here, after the induction protocol, stimulated cells in the low density experiments had persistently biased activity for up to 24 hours. And I'm not showing this here, but also they had a significantly higher rate of new place field formation at that location than one would expect from chance. So to understand at a finer grain level why some stimulated cells form new place fields and others don't, we separated the stimulated cells based on outcome. So we first found that induced cells had much larger responses to stimulated cells that did not induce place fields. And interestingly, we also found that induced cells tend to be in locally less dense regions of the field of view. So for each population, we can ask what the average number of other stimulated cells was in an expanding radius around the cell. And we found that in a neighborhood of up to about 15 microns, induced cells had fewer stimulated neighbors than non-induced cells. So if we look specifically at the stimulation response of cells with at least one neighbor in a given radius, we again see a particularly large separation in the groups at that same spatial scale. So together, these results support maybe a local winner-takes-all model in which only the strongest bursting cells in highly active neighborhoods can form new place fields. So to find further evidence of recurrent inhibitory mediation of place field formation at these spatial scales, we coupled the same sparse opsin expression strategy now with 2-photon targeted stimulation of opsin positive neurons. So with these strategies combined, by spiraling a laser over light responsive cells, this yields a spatially restricted stimulation response in the targeted cell with adjacent cells either slightly inhibited or totally unresponsive to stimulation. And as expected, we found that this protocol, which is different only in the method of light delivery, also induces persistent place fields, specifically in the stimulated cells, with no bias in the unstimulated populations. When we instead couple this 2-photon targeting with wide or dense opsin expression in all pyramidal cells, you can see that 2-photon stimulation results in responses with a wide halo of cells around the target cell due to the presence of overlapping dendrites and axons and the non-negligible radius of the 2-photon cross-section. So we wanted to know how this local increase in activity might affect BTSP induction efficacy. And then here we're also going to target an inhibitory dread, so it's activatable by systemic CNO injection to all local interneurons, which is going to then allow us to temporarily slightly suppress inhibitory activity. So with this strategy, we find that dense stimulation with inhibition intact, so that's here in blue, totally disrupts BTSP efficacy with no bias in stimulated cells towards the stimulation zone. So locally stimulating even very small single-digit numbers of cells prevented BTSP induction. Importantly, though, relieving inhibition here in yellow via CNO dread activation partially rescues BTSP efficacy in a manner that is again dependent on the number of cells stimulated, suggesting that this weak relief of inhibition is still not enough to overcome the strong recruitment of local interneurons in higher density cases. So these manipulation results really strongly implicate local inhibition in mediating BTSP induction. So what is the impact of this type of local circuit control over place field formation on spatial learning behaviors? So as I alluded to earlier in the talk, previous work from our lab and others has used a goal-oriented learning paradigm to probe spatial learning. So in this task, a mouse finds the location of a series of fixed hidden rewards, and then we can quantify the mouse's learning by looking at the fraction of licks near the reward zone. So where a more uniform exploratory licking behavior gives way to a very specific licking at the reward zone. And the key finding here was that after reward switch, place fields tend to overrepresent the location of their new reward zone, as can be seen at the bottom left. And the extent of this overrepresentation is in turn correlated with performance. So we therefore wondered if increasing the place field formation near the future reward zone could bias behavior towards that location and therefore increase learning performance. So to this end, we bilaterally injected Cre-dependent carmine alongside tamoxifen-inducible Cre throughout a large extent of hippocampus and implanted optic fibers over CA1. The mouse then underwent a modified goal-oriented learning paradigm in which during the last session of condition 1, they underwent the induction protocol at the future location of reward zone 2. And then on the next session, the reward zone is switched and the mouse has to search for the new reward zone as normal. And so we can look at this behavior before and after inducing opsin expression with the tamoxifen injection, giving us an internal control for each mouse. And incredibly, we found that sparse induction of place fields near the reward zone was sufficient to improve spatial learning performance. So as you can see, after the induction protocol, mice expressing the opsin had a very large and significant increase in performance, finding the reward at the stimulation location, which persisted out to the next day. Importantly, though, prior to opsin expression, so that's here the control in black, the induction protocol has no impact, suggesting that this could not be a result of light cueing or any other artifact. At right, when we merge the sessions for each conditioning group, this clearly demonstrates a specific performance boost for the opsin positive group after the induction protocol. So in data I don't have time to show, this seems to be due to differences in learning rates. And together, this all suggests that preferential place field formation is enough to causally impact spatial navigation behaviors. In summary, we first found that we could optically induce BTSP in single cells, and we argued that recruitment of local feedback inhibition could be responsible for gating BTSP at higher population activity levels. To this end, we directly demonstrated that local interneurons scale their activity with pyramidal cell stimulation density, and that there was a strong inverse relationship between stimulation density and the efficacy of BTSP induction. Using 2-photon targeted stimulation, we showed that we could, to some degree, rescue high-density BTSP induction through partial silencing of local interneuron activity, strongly implicating local feedback interneurons in the scaling of BTSP induction rates. And then finally, we took advantage of these results to overrepresent place fields at a reward location and found that we could bias learning at that location, demonstrating the importance of existing place field representations in driving spatial navigation. So with that, I'd just like to acknowledge all the other folks that worked hard on the project, so especially my mentor Attila, 2 other scientists in the lab, Mickey, with whom I did all the single cell work, and Mohsin, with whom I did the target stimulation and goal-oriented learning work. Thanks to a number of others, to everyone funding the work, to all the organizers and all of you. And yeah, I'm happy to take any questions now, or feel free to follow up via email. So thank you so much.
Thank you, Sebi. Thank you on behalf of 1,108 active viewers. We have a few questions, probably more raining in. The first one is, do you think the results depend on if you stimulate the soma as opposed to the apical dendrites, or the inputs targeting those compartments?
Yeah, great question. So here the opsin, I mean, especially for the, in the wide field WD case, we expect that the opsin is going to be expressed throughout the cell. So we expect that we are actually stimulating the entirety of the cell. So I suspect in that case, you know, so we don't necessarily think that this has to do with, and in the 2-photon targeting case, we are basically very specifically targeting the soma, and we see very similar results between those two. So it doesn't seem to be a compartment stimulation specific effect. It seems that as long as the soma, yeah, we suspect that it has to do with apical bursting, but we don't have direct evidence of that, I guess is the best thing to say.
All right, another question by Roman Huzar. Can you speculate on which mechanism BTSP or pre-existing network constraints occur in spontaneous remapping? Beautiful work linking 2 contradicting results.
Oh, thank you, Roman. I think it's very hard to say. I think, you know, based on your results, I think it's very likely that existing network dynamics are playing the major role in remapping between contexts, but I think it's likely a combination of the two, frankly. And I think, yeah, a whole lot of work needs to be done to, which, you know, work that's ongoing in our lab as well, to actually sort of try to pick out how these two different sort of forms of plasticity perhaps are what the interplay there is.
Maybe 2 more questions very briefly. Number 1, could you resolve what kind of interneuron was involved by Eliv Mueller? And then what happens if you stimulate neurons that are not at the reward location? Does that interfere with learning? Does that confuse the animal?
Oh, interesting. Yeah, so regarding the first question, we don't know. We suspect that they are either OLM cells or bistratified cells. It presumably needs to be some local, some interneuron that receives local axon collaterals from C1 pyramidal cells, and presumably is also projecting down to the apical dendrites. But we don't have any direct evidence right now as to what interneuron subtypes are actually responsible for this. And yeah, we're very curious to find out. And sorry, what was the second question? Oh, yeah, we have not done that experiment. I think that'd be really interesting. I think recent work from the Heuser lab might speak to that perhaps better, where they're stimulating at either the reward, you know, start cells versus reward cells tend to elicit different behaviors in the mouse. I suspect that we would perhaps degrade performance, but I'm not totally sure. Yeah, that's a really interesting experiment.
Thank you, Sebi. This concludes your show, your performance, as it's now 6:20.
Okay.
Sanjukta, can you come online? The Heuser lab there was our keyword because the next speaker, Sanjukta Krishnagopal, is presenting work from pretty much a team of all-stars, including Michael Hauser, but also Claudia Klopath and Peter Lathan. So without further ado, Sanjukta, the stage is yours.
Great. All right, so hello everyone. My name is Sanjukta Krishnagopal. I'm a postdoc at the Gatsby Computational Neuroscience Unit, UCL. And today I'll be talking about dendritic gated network inspired alternative team here at UCL periodage. Right.
Sanjukta, might be me, but we're having a little problem with your upload speed, so it might be worthwhile turning off your camera because I can't hear you.
I see. All right, let me try to turn off my camera.
All right.
I hear an echo. All right, great. Okay, so hopefully everyone can hear me now.
This is much better, thank you.
Okay, so basically, I think, well, great. I think it's well accepted that the brain happens as a consequence of changes in synaptic weights. And these synaptic weight change is still a bit unknown. And so if you ask machine learning people, they'll tell you the answer is backpropagation, where some input goes through the network through several layers all the way to the output.
Sanjukta, we're still having issues with your audio. Any
layer
chance
by
you can
layer.
move a little closer to your router?
Okay, let me try that.
I apologize on behalf of
All right, how about now? Sorry about that. It should be working now. Any better?
We'll try. Let's go.
All right. Okay, so basically, backpropagation. It's basically an input that passes through several layers, goes to the output, computes an error, feeds it all the way to the neurons, and this simply is not biologically plausible. So a quick fix to this problem, where each neuron, irrespective of what layer it's in, the job is to directly predict the target. So the target here is fed back into each neuron, and they don't have to do this forward pass, backprop business. They can sort of almost instantly, change their synaptic weights. However, just local learning, just implementing straight up local learning doesn't quite work. And so in the hopes of getting something to work, I'll introduce our model, dendritic gated network. And this is basically, so this figure on the left here is something everyone's familiar with. Dendritic gated networks has each neuron here, each neuron has these several dendritic branches. These branches receive inputs from the previous layer here, so all the weights from these layers synapse onto these branches. So here are 3 branches in this particular example. And we implement something called gating on these branches, which turns these branches on and off. So for example, this branch in green here is on, this is the gate function, this one's off, these 2 in red are off. This is that the weights that are involved connected to this branch here learn, and they are actually updating, information is passing through them, whereas these 2 branches, nothing's flowing through. And this is local not working to quite some degree. And this is actually inspired by the ability of biological interneurons to gate activity in the brain of single dendrites. And so we have this gating function on each of the dendrites of the. All right, so how exactly does this gating work? So let's just focus in on this branch over here. And the gating is input dependent. So the input goes into this gating function. And the gating function is gated on and off depending on the input. So let's consider a really simple 2D input, x1 and x2. And we basically draw random hyperplanes across the input space, and in 2D that basically just becomes random lines across the input space. And if the input comes in this region here, gate is open, information is flowing through, learning is happening, all those weights, and in this region here, no learning weights. Let's extend this to B1, B2, B3, and let's consider this example input here, this blue dot here, where X1 and X2 fall here. This branch B1 is closed, B2 is open, B3 is open. And again, what that means is that weights are flowing, are learning through this for this specific input. So there's learning happening along these branches, whereas there's no line to this branch B1. For a different input here, we have a different set of branches that are active. So B1 and B3 are active, and B2 is not. So basically, through this type of mechanism, through this type of gating, it results in a different subset of weights being involved in learning for different inputs. So each input turns on and off a different subset of branches. And that actually does really well for solving this problem of local learning not working by itself. So just kind of bringing it all together, dendritic-gated networks is this biologically plausible learning inspired ability of interneural, this kind of inhibition in the brain. It's actually different what people in machine learning or computational neuroscience are typically used to features, where the input goes in and gates each of the branches. And this is what causes it to be resisting old learning. So if it's learning a bunch of stuff, in the past, and it's learned something else more recently, it tends to not forget what's learned previously. Also, local learning, where the target gets fed to each layer. This makes it extremely data efficient because it doesn't have to do this whole backprop business, and the loss becomes, here it's just minimizing whatever the output of each neuron is and the target, some distance of the output and the target. And so loss becomes convex with respect to each way learning. So let's talk a little bit about forgetting of old tasks. And here I'm going to talk about a sort of toy example that shows you exactly why the DGN tasks. So here's an example input. Think of it as like an image, 2D input, X1, X2. And it has these red and blue regions that are linearly inseparable. And so we compare our DGN with an MLP, a multi-layer perceptron, in separating the red and blue regions. And so the MLP, when it's trained on this task and then tested again on this task, it's very good at figuring out which region is red, which region is blue. These decision boundaries here tell you that the MLP classifies this entire region as this entire thing is blue, and so on and so forth. And it's great. However, task B, which is almost orthogonal to task A, and then we train the MLP on first and on task B, and then test it on an old task, task A. And the MLP does pretty well after being trained on both tasks on task B, but now it's changed its decision boundaries to match that of task B. So when you test it on task only one task before, it already does much poorer, it forgets quite a bit. And train on task A, test on task A, still does pretty well. Whereas if you train task A, train task B, and test again on the task, it remembers quite a bit more. So the accuracy is still coming in at 91%, which is great. So the DGN is much more resilient to forgetting old tasks. Now, this was kind of a toy example, but we're going into detail. We also on benchmarked MNIST, continuous learning task. And we tested it on 10 different tasks about changing random permutations of the pixels, and tested on task 1, the first task, all the way in the beginning, significantly better well. All right, so this dendritic-gated network is very much inspired by the cerebellum, and I'll tell you a little bit more about how they're related. So the cerebellum for a long time was thought to be a one-layer network, but recently there's evidence that it's actually a two-layer network. So there's the parallel fiber Purkinje cells, which is the first layer, parallel fiber inputs coming in here in blue, and that's on bunches of the Purkinje cells, which you can think of as the hidden layer. And then there's a second layer, so Purkinje to the cerebellar nuclei, and then the cerebellum, spits out the output that goes to the motor regions. These molecular layer interneurons in pink are known to play an inhibitory role in the brain on the dendritic branches of the Purkinje cells. And they play a role that's pretty similar to gating in our framework. And lastly, is carried back by the climbing fibers, carried to the output layer, also to the hidden layer, the Purkinje cells. Like we do with local learning, we send the air signal back to the hidden layer as well. Now we want to test this on a task that's a benchmark cerebellum task. So vestibulo-ocular reflex is a very popular benchmark task people use for cerebellum studies. And basically what it is animal moves its head while focusing on an object. Its eyes move in the opposite direction. And so you can think of it as, focusing on an ice cream. The object can move in the direction of your head motion or in the opposite direction of your head motion. And then your eyes have to sort of move a little more to compensate for the object moving as well. So this here is just moving the same direction. It's a gain down stimulus. Opposite directions, a gain up stimulus. Cerebellum is critical for VR gain adaptation. So we try to model this type of VR. So we give it head signal, head velocity, which is just a random aperiodic signal that looks kind of like this. And then we map the cerebellum directly onto the DGN. So the cerebellum had these parallel fiber inputs. And then in the first layer, these parallel fiber inputs each has a different delay. So this is just the signal ST with a different delay. This delay represents time it takes the signal to go from your retina to the parallel fiber. And then they synapse onto the dendritic branches of the Purkinje cells. And then the Purkinje cells go to the CBN. And so the goal is to predict the modulated eye velocity. So this minus gain times S of t. And so as a baseline, we use an MLP, which is for a two-layer cerebellum task. It's like the only baseline. So it seems pretty reasonable to use this. All right, so this is a pretty cool task from a machine learning perspective. And so both MLP and DGS do it well. But if you look under the hood, here I'm plotting 5 different Purkinje cells, so 5 different hidden neurons. And I'm going to zoom into one of them. So each hidden neuron has a lot of branches. So each curve represents one branch of the Purkinje cell, and several parallel fibers are synapsing onto this branch. So each, each dot here, which are connected, is the weights of these parallel fibers that are synapsing onto a specific branch. And they're sorted on the x-axis according to the parallel fiber delay. So anyway, without getting too technical, you can see that these weights here have a very pattern in delay. So the weights synapsing onto the Purkinje cells are smooth. Whereas if you do the same thing for LP, there's no such smoothness. And so the DGNs learn kind of a smooth, more sort of plausible vision. All right, we also wanted to the dendritic gated network. We predict that the little hyperplanes or the lines that determine whether a gate is on or off are fixed over learning. So these don't change over learning. And to do this, we ran an experiment. To test this, we ran an experiment of 2-photon calcium imaging in a mouse that's learning the board. Without getting into too many details, beautiful data. And here's what the results look like. I tried to measure, as the mouse was learning over 125 trials, we tried to measure correlation in the activity of the molecular interneurons, which represent the, as well as these climbing fibers, which represent the air. And as you can see, these climbing fibers, as the mouse is learning, the air is down, correlated, but it's are relatively quite highly correlated, which is a feature of the DGN. These gates remain unchanging. So these experimental results are quite consistent with our model. So in conclusion, I want to say we introduced these dendritic gating networks, which are radically different and a biologically plausible learning role. They propose a new role for gating that interneurons, a new role for interneurons as gating functions in the cerebellum. We show that DGNs are resistant to forgetting, unlike sort of MLP type machine learning architecture. We map it quite directly to the cerebellum, all these similarities with the biological cerebellum, and we provide experimental support which suggests that our model is consistent with what we know of cerebellum and interneurons in the cerebellum. And that brings me to the end. Thank you so much for your time. I want to thank everyone that I showed right in the beginning that played a part in this project, and I'm happy to take questions.
Thank you, Sanjukta. I hate to say your upload speed teetered on the brink of disaster. It was at times spotty. Forgive some of the questions if they've been mentioned before, but I think we have quite a few. So one by Cristina Savin is, does the gating control just plasticity or also the feed-forward flow of information?
So that's a great question. So the gate. Sorry, I can hear some feedback.
That's me.
Yeah, so the gating does control the feed-forward flow as well. So when a gate is off, nothing flows through, nothing is learning, nothing's happening at all. So that gate is simply off. But the interesting thing is that different gates are on and off for different inputs. And so certain weights are only responsible for learning a part of the input. And so this is what enables it to do better.
And how do you know which dendrites to gate? Is that predetermined by you, or is that part of the learning process?
Right, so that is predetermined, and the gating of dendrites, basically going back to that slide, going back to that slide where the input is divided into regions. The division of the input into regions, these sort of lines that you saw that divide the input, they're randomly picked. And so as long as you have enough neurons, you can randomly pick these lines to split up the inputs such that each input has a unique subset of dendrites that is responsible for learning specifically that input.
Jan Drugovic wants to know if, given that all neurons could in principle receive the same feed-forward and feedback information, whether you could elaborate on what you gain through multiple layers.
So each layer, sorry, I hear some feedback again. So each layer, so you're right. So with local learning, the advantage of different layers kind of goes away to a small extent. But you can, for example, in higher up layers, the information from lower layers is aggregated such that each neuron in a higher up layer can do, at worst, at least as well as the best neuron on the previous layer. So this gating mechanism, which controls only certain parts of activates only certain branches of the input for a specific input, then goes on in the next layer to improve on that, such that the input space is now, for example, if you take the input space, and in a previous layer, you split it up, and one set of weights is learning one half of the input space, and another set of weights is learning another half of the input space. The next layer is then able to aggregate this information. And so the next layer is able to learn sort of combine information from both sides of the input. So it's learning a little bit more than just one region of the input.
Thank you. Anna Kuchirata wants to know whether the number of gates determine the capacity of different tasks that can be learned, or what else could determine the capacity of the number of tasks you can learn.
Yeah, so that's a good question. Definitely you need a minimum number of gates per input such that the input is adequately represented. So the number of gates do play a role, but as long as your network is above a certain threshold, becoming too large is kind of giving you, it's extra computation without much benefit, in my opinion. So as long as you have enough representational power in your network, you should be able to learn simple tasks. And so actually this gating takes linear functions and is able to learn nonlinear functions by combining linear pieces of different functions. And so if you had a highly nonlinear function that you were trying to learn, you would have to have a larger number of piecewise linear sections that each neuron is learning. And so that's kind of a constraint on how difficult of a task you want and how big of a network you require to be able to solve that. Thank you.
Before I ask you the last question, or I guess it's already time's up, I suppose we're at 20 or at 40 exactly. So Sanjukta, I'm afraid I have to cut off here. There was an interesting question about recurrent networks and whether you could replace backpropagation through time, but I think we're going to have to discuss that in the social sphere of this meeting, if there is one. So thank you, Sanjukta. I'm going to have to ask Naixin to come online, and thank you again for your talk, Sanjukta. Maybe sometime in person. Bye.
Yeah.
All right. Our last scientific presentation of the session by Naixin Ren. It's my special pleasure to introduce her because Naixin has also been a student at what I think is probably the best by a large margin summer school on the planet in Cape Town. And I should also say that applications for that summer school will open soon because we're expecting the pandemic to be over by January 22 when that summer school is being held in the global south, so in the summer down there. Also funded, by the way, by the Salmons Foundation, which will be speaking after Naixin's talk. Naixin is at the University of Connecticut in Ian Stevenson's lab, and she will talk about predictive fluctuation in synaptic strength due to the natural variation in presynaptic firing rate. So without further ado, Naixin, the floor is yours.
Okay, thank you so much for the introduction, and let me share my screen. Okay, thank you for the introduction. I'm Naixin Ren. I'm a fourth-year PhD student at University of Connecticut, and today the topic I would like to talk about is the predictable fluctuations in synaptic strength due to natural variation in presynaptic firing rate. So based on many previous in vitro studies, we have learned how synaptic strength can vary with presynaptic spike timing due to short-term synaptic plasticity. For example, for the synapses with short-term depression, they should be weaker when the presynaptic firing rates are higher, and they should be stronger when the rates are lower. For example, this is a picture adopted from Abed et al. In 1997. So in their study, they gave the presynaptic neuron stimulation at different frequencies, and they found the response in the postsynaptic neuron decreases as the frequency increases. And on the right, here's an example of facilitation. So you can see the response of the neuron are higher during high frequency stimulation. So these are the examples from in vitro studies. It is still unclear how the natural fluctuations in presynaptic firing rate affect synaptic strength in vivo. So here in this study, we would like to use an in vivo data set to study the following questions. So the first would be, how do the presynaptic firing rate and also the synaptic efficacy fluctuate over time? So in particular, we would like to study the scale of the fluctuation. And then we're interested in the relationship between the efficacy fluctuations and the natural variation in presynaptic firing rate. And we would like to see whether STP is involved in this process. So the data set that we use in this study is from Ali Institute. So here we use a large-scale in vivo actual cellular spike recordings from the Ali Institute NeuroPixel data set. So here this is a drawing of the probe that we use. So since the probe is pretty long, so here in this data set, the spiking activities were not only recorded in the mouse visual cortex and also in some deeper areas like subcortical visual areas and hippocampus. And during the recording, each mouse was exposed to a standardized set of visual stimuli. And here, there's one thing that I would like to mention. So since here we're using actual cellular spike recording, so we don't have any access to potential recurrence. So we don't have anything like EPSC or EPSC like we usually see in vitro study. So here for the following analysis, all the analysis are based on spike data. So since here we have the spike data, the first thing that we need to do is to detect synapses from those spike data. So here we detect the putative excitatory synaptic connections using cross correlations or cross parallelograms, CCG, between the neuron pairs. So on the right, you can see some examples of the CCG of the putative excitatory synaptic connections we detected from the data set. And if you're not familiar with CCG, it is generated using the spike trains of two neurons. One we call it as a source neuron, and the other we call it as a target neuron. And you can think the CCG as a histogram of the spikes in target neuron at different interspike intervals between the source neuron and the target neuron. So for example, if you see a peak that's around, let's say 2 milliseconds here, then this means that the target neuron is more likely to fire 2 milliseconds after the source neuron fires. And that may indicate that there is a putative excitatory synaptic connection from the source neuron to the target neuron. So this is how we detect synapses using the spike data. And then to estimate the efficacy, here we get an extension of the generalized linear model to the CCG. And here, this model is developed based on the model that we published last year. So briefly, in this model, we divide the CCG into two parts. One is the slow fluctuation, which we fit using some slow basis functions. And the other is the fast synaptic effect, which we fit using a fast alpha function and scaled by this W. And this W is the synaptic weight in this model. So here on the right, you can see the model fitting results on some CCGs, which are the red lines. So you can see this model fitting is able to successfully capture the shape of the cost program. And then in order to calculate the efficacy, we not only calculate the model fitting using the full model, we also calculate the model fitting when we set this W to be 0. So in that case, there's no alpha function in this model fitting. So the model fitting only captures the slow fluctuation. So in that case, when we calculate the efficacy, we calculate the difference between the red line and the yellow line, and then divide that by the number of presynaptic spikes. So in that case, you can think efficacy as a ratio of the extra spikes brought by the presynaptic spikes to the number of presynaptic spikes. So again, since we're using spike data, so here this efficacy is not the same as the traditional efficacy that we usually talk about in in vitro studies, which are EPSP or EPSC. But you can imagine here this efficacy is affected by EPSP or EPSC. Okay, and now since the goal of the study is to study the fluctuations in efficacy and presynaptic firing rate, so here for each connection, we divide the pre and post synaptic spike trains into 5-minute windows with 80% overlap and estimate the efficacy and also calculate the presynaptic firing rate for each window separately. So here shows an example putative excitatory synapse within CA1, and here shows the traces of the presynaptic firing rate and efficacy, and show the fluctuation of them on a minute time scale. And here are also two example windows. One of them is with high efficacy, and the other is with low efficacy. And here, in order to quantify the fluctuations, we calculate the coefficient of variation for these two variables. So here, coefficient of variation is standard deviation divided by the mean. So this means that for this particular connection, the presynaptic firing rate fluctuates at plus minus 20% of the mean over time, and the efficacy fluctuates at about plus minus 78% of the mean. So we calculate the CV for these two variables for all the putative excitatory connections that we detected in the dataset, and here shows the histograms of the CV for presynaptic firing rate and efficacy. So we can see that for a typical excitatory synaptic connections, the presynaptic firing rate would fluctuate at about plus minus 30 or 33% of the mean, and the efficacy would fluctuate at about 70% of the mean. So this basically shows that for most of the connections, the presynaptic firing rate and the synaptic efficacy fluctuates a lot over time. Okay, so now we've learned on what kind of scale the fluctuations happen, and the next step is to study the relationship between the two fluctuations. And from the data set, we discovered 3 different kinds of relationship between the fluctuations. So the first one is more like a depression kind of relationship. So here the blue scatter plot is between efficacy and the log of the presynaptic firing rate. And we can see that as the efficacy, so the efficacy decreases as the log firing rate increases. So this is a depression kind of relationship. And here this R square, this R is from Spearman correlation. And here this one is an example of a facilitation case. So we can see efficacy increases as the log firing rate increases. And here this is a case when there's just no clear linear relationship between efficacy and log firing rate. So from this data set, we discovered this 3 different kind of relationships between the fluctuation in efficacy and fluctuation in preset firing rate. And here what I'm showing is just a description of the relationship. And on the next step, we would like to find the explanation for this relationship. And we would like to see whether STP serves as a mechanism under the relationship between efficacy and presenting firing rate. So here we developed a GBLM of STP, which is the generalized bilinear model of STP. So using this model, we can estimate the time-varying synaptic effect by modeling the post-synaptic firing rate. So here, the post-synaptic firing rate is modeled as a combination of the 3 components. So first is the baseline, and the second is the post-spike history effect, and the third is the synaptic coupling effect, which varies a long time. And here, this WT varies as a function of the pre-synaptic interspike intervals, and this is the STP part of the model. So here shows an example of a depression synapse. So here, this is the time varying trace for WT, and you can see it changes as a function of different pre-synaptic interspike intervals. And here on the bottom, this black line is the coupling without STP. So this is the coupling if WT is set to a constant at 1. And here, this red line, this is the modified coupling with STP. So, so this is the coupling when we set WT varies as a function of ISI. So in this case, since this is a depression synapse, so you can see the coupling with STP is smaller than the coupling, the unmodified coupling without STP. So here we use this model of STP to fit all the connections, and we would like to see whether the estimated coupling using this model, whether that is able to reproduce the relationship between efficacy and firing rate. And here again, let's take a look at this example of the depression case. So here on the right, there's this green line shows the estimated GBLM coupling, and we can see the GBLM coupling is able to reproduce the fluctuation in efficacy. And here, the green scatter plot is between GBLM coupling and log firing rate, and we can see it is able to reproduce the depression kind of relationship between efficacy and firing rate. And here, this black scatter plot is between GBLM coupling and efficacy. And we can see GBLM coupling is able to reproduce fluctuation in efficacy. And here shows an example of a facilitation case. And we can see, again, GBLM is able to reproduce this facilitation kind of relationship between efficacy and log firing rate. And again, GBLM coupling is able to capture the fluctuation in efficacy. And this is the example when we don't see any clear relationship between efficacy and firing rate. And again, we don't see any clear linear relationship between GBLM coupling and firing rate. However, in this case, GBLM coupling is still able to reproduce the fluctuation in efficacy. And of course, there are other connections when we don't see clear linear relationship between efficacy and firing rate, and we don't see GBLM coupling reproducing the fluctuation in efficacy. Okay, so for a summary result, so for the current stage, we manually selected connections with short latency and short time constant in the peaks in CCGs. So in that case, we know for sure that these are strong and clear connections. And for now, we've run GBLM on 224 connections. And in all these connections, the relationship, so we calculate the mean R square between efficacy fluctuation and pre-synaptic firing rate fluctuation. And the mean R square is about 0.12. But you need to note that there are some of them are not, some of the connections there's no clear relationship between efficacy and pre-synaptic firing rate. And we found that in those 224 connections, more than 64% of them are significant. And we are also interested in whether GBLM coupling is able to reproduce the fluctuation in efficacy. And here, the average R-square between these two is still at the same level. And again, more than 63% of them have a significant correlation between efficacy and GBLM coupling. So as our conclusion, so we found that for individual connections, the synaptic efficacy often varies substantially over time, and the efficacy fluctuation mirror fluctuations in the presynaptic fire rate. And for many connections, we found that the GBLM of STP can accurately reproduce the observed efficacy fluctuations, and this suggests that these fluctuations can be at least partly explained by a cumulative effect of STP. Okay, so I would like to thank my advisor, Dr. Ian Stevenson, who helped me a lot in this project, and also thank for Dr. Abed Ghanbari, who graduated from my lab, and he developed the GBLM, and also thanks for Al Institute for providing us this data set to analyze, and also thank NSF for providing us the funding. So that is it for the talk. Thank you so much for listening. Please let me know if you have any questions.
Lovely. Thank you, Naixin.
Thank you.
We have a few questions. Maybe first, did you have any candidates that you could not explain by just short-term plasticity alone? Did you see any footprint of longer-term plasticity happening in your data?
Yeah. So I think I hear echoes. Yeah. Thank you. So even in the case that I'm showing now, you can see that STP is not enough to reproduce all the fluctuations in STP, in efficacy. So we would expect that LTP is playing some kind of role in this fluctuation. And in our future plan, we're planning to incorporating LTP in our model to see what kind of contribution LTP could make in this for this relationship between efficacy and fire rate.
Okay, thank you. Peng Hao Shan Tan is asking, I'm wondering if the CCG might capture contamination from spikes that are improperly sorted and belong to the same neuron instead of connected spike pairs.
Yeah, that's a good question. So one of the reasons that we use this data set is because they use KiloSort2 as the algorithm when they do spike sorting. So the good thing about KiloSort2 is that it has the correction for drifting spike problem and also spike shadowing problems. So we assume that KiloSort2 should have a better performance than some traditional spike sorting algorithms. So I think the KiloSort2 algorithm itself helps us resolve some of the problems.
Thank you. Trang An Nguyen, approximately how many putative synapses could you identify? And from recordings of how many neurons, I think you answered that partly. How many excitatory and how many inhibitory synapses? I didn't mention that in my talk. So our analysis is based on 10 data sets from this NeuroPixel data, and each data set contains from 300 neurons to 900 neurons. So from all these neurons, we detect about 900 putative excitatory synapses. It's just for now, we only run GBLM on part of the neurons. And for this stage, we only focus on excitatory connections. So we haven't do any analysis on inhibitory connections. And I do see that there are a lot more excitatory connections than inhibitory connections. We have time for 2 more questions, maybe. Do you consider that confounding upstream neuron may reliably activate both neurons that seem to communicate with each other? So the common source problem. Yeah.
We think that might be one thing that may be affecting the shape of cross programs. So at least for now, for all the strong connections that we're analyzing now, that's 225 connections. We think we're pretty sure they are strong, clear connections that's not affected by common input problems. And for all the connections that we detected, when we were detecting the connections, we tried to exclude the connections that seems like they have a common input which cause a peak or maybe a trough right under the center. Yeah.
All right. Last question, maybe. Now I lost it. There it is. Is it possible that the apparent coupling strength depends on postsynaptic firing rate?
Yeah. I'm not sure this coupling strength that you are talking about, whether you're talking about the GBLM coupling or the efficacy. I think for some synapses, it is possible that postsynaptic firing rate is affecting efficacy. Actually, I think I can show some examples here. Yeah, so like for this one, I plot the postsynaptic firing rate here. So this is the case that you can see, you don't see any clear linear relationship between efficacy and postsynaptic firing rate. And like this one too, you don't see any clear relationship between post firing rate and efficacy. And also this one, you don't see any clear relationship. So I think for some of the synapses, postsynaptic firing rate is not affecting the efficacy a lot. But I want to say that we do see some cases when we see the postsynaptic firing rate is affecting efficacy. So in that case, I would like to say that there are some cases when postsynaptic firing rate is affecting that. And we are planning on improving GBLM to make that incorporate the fluctuation in postsynaptic firing rate to see if that improves the performance.
Thank you, Naixin. I think that was the last question because we're at the end of your time slot. Some of you asked what the summer school was that I mentioned. It's the Eero Simoncelli Computational Neuroscience Imbizo in Cape Town. Imbizo is a word for a gathering to share knowledge in Zulu. So it's not strictly speaking a summer school, but a gathering to share knowledge. And as the name implies, it's largely sponsored by Simons and by Eero. Simons is the foundation that will also now speak about Bridge to Independence Awards, because the Simons Foundation has been an active participant in the funding landscape for neuroscience in general and has been trying to do as much good as possible, I think, thanks to the generous support by James Simons. So Laura Long and Alice Luo-Clayton maybe come online and share your wisdom about how to facilitate the transition from postdoc to junior faculty.
Great, thanks a lot.
Hi, how are you? Thanks for having me.
Okay. Thanks a lot, Tim, for the introduction, and to the Cosyne organizers for having us, and to all of you for joining us. My name is Laura Long. I'm an assistant scientist at the Simons Foundation, working with the Simons Collaboration on the Global Brain. I'm happy to have my colleague Alice Luo-Clayton from SAFARI, the Simons Foundation Autism Research Initiative, here with me today to tell you about our Bridge to Independence Awards available through the Simons Foundation. First, a little bit about us. We are a private foundation whose mission is to support basic math and science research. The foundation does this in two ways: through external grant-making groups, which include SAFARI and SCGB as part of life sciences, and the Flatiron Institute, which conducts in-house research. Today, two Simons Foundation programs, those that we represent, of course, SCGB and SAFARI, are currently offering Bridge to Independence Award programs. I'm going to share about SCGB's and hand it over to Alice to talk about SAFARI's. The Simons Collaboration on the Global Brain, SCGB specifically, funds systems and computational neuroscience, so is very excited to always participate in the Cosyne community, with the aim to expand our understanding of the role of internal brain processes in the arc from sensation to action, to help us understand the nature, role, and mechanisms of neural activity that produces cognition. With this Bridge to Independence Award, we seek to fund scientists doing work consistent with this mission, ideally investigating large-scale circuits at high spatial temporal resolution to understand neural dynamics and coding. So about the award itself, the Bridge to Independence, or BTI program, engages talented early career scientists by facilitating their transition to research independence and providing grant funding at the start of their professorships. The award is aimed at scientists who are currently in training positions but intend to seek tenure track research faculty positions during the upcoming academic job cycle. Selected fellows will receive a commitment of $495,000 over 3 years, which is activated upon assumption of a tenure track research professorship. Importantly, although the eligible applicants must be currently in a training program, the BTI award itself is not a training fellowship, but a research grant to newly appointed faculty. The program selection process is specifically designed to enhance a fellow's job prospects by providing a letter that specifies our financial commitment to the proposed project once the BTI fellow has secured a suitable faculty position. The grants are awarded through a multi-stage process. The first stage, in blue, Open Now, is a competitive process where applications are reviewed by the SCGB Scientific Staff and Review Committee, and fellows are selected at the end of stage 1. In stage 2, in orange, upon notification, fellows have 18 months to secure a tenure-track professorship at an academic institution, at which point they'll submit a second stage approval application. We also encourage fellows to be in touch with staff during the job search process, as we're happy to provide advice and insight and make sure that, and coordinate to see that the opportunities you're interested in are aligned with and able to be and consistent with activation of the award. In gray, once we receive the approval application, it's a non-competitive internal review, just intended to confirm that the fellow secured a position and adequate institutional resources to accomplish the Bridge to Independence research proposal. In terms of eligibility, applicants must hold a PhD, MD, or equivalent degree, must currently hold an non-independent mentor training position, so a postdoctoral fellowship or equivalent, should plan to land a faculty job in the next 18 months, and SCGB does not have any length of training or citizenship requirements. I will note that applicants may not hold an award of similar scope, for example, a K99R00, and may not have officially accepted a formal faculty job offer at time of application. Also a note on award activation, so not eligibility to apply per se, but when a fellow has then secured their job, we do not have any country restrictions on where that job needs to be. All applicants that fit these criteria are welcome to apply, and SCGB especially encourages applications from diverse candidates. In terms of the timeline, applications are open now and due April 5th. Finalists will be notified on May 18th and scheduled for virtual interviews on May 27th, and then selected fellows will be notified by the end of June. You see here contact information for our administrative team in terms of issues with submitting the application, and then for the scientific team where we are happy to answer questions, and a QR code to the application itself. So we hope to see applications from many of you. Happy now to hand it over to Alice to tell you about Safari's program. And I also want to note that while SCGB is starting this program up and offering this for the first time this year, Safari started their BTI in 2015 and have been exceptionally generous with advice and insight on how to run a program that's been as successful as theirs, and we're excited to emulate it.
Thanks so much, Laura. Can everyone hear me? Thank you. Thanks to Laura, to SCGB, and to Cosyne for allowing me and the rest of Safari to talk a little bit about this really exciting program. So I wanted to start a little bit about Safari itself, because most of you are probably, I assume, not as familiar with the Safari portion of Simons Foundation. So Safari does stand for Simons Foundation Autism Research Initiative. We were actually started back in 2007, and we are the largest private funder of autism research in the US. Due to our very generous donors, we've always had a really great operating budget, and this year it is now at $100 million. So we advance science through a variety of different ways. Most well known is our grant funding, but we also know that giving the community access to great resources and to data is also a critical aspect of it. So we put an enormous amount of effort into developing high quality, well-characterized patient cohorts with accompanying genomic and phenotypic data. And I think importantly for this particular community, we've made a huge investment in creating animal models as well as cell models. So this includes mouse models, but also rat models that I've been personally involved in, and zebrafish models as well. So if you could just click to the next one. So there's lots of ways that people contribute to, oh, sorry, can you go back one more? Thanks, Laura. So there's lots of ways that one can contribute to the autism community. For Safari specifically, our scientific mission is really focused on the biological underpinnings of autism. And with that, we really do support the sort of entire breadth of biological sciences, from genetics through to molecular mechanisms, circuit and systems, and clinical science. So now on to the BTI. As Laura mentioned, I happily started this in 2015, and it's been going extremely well. It continues one of the longstanding ethos of Safari itself, which is that we want to bring in new scientists into the field. We know that tackling autism really requires input from many disciplines and many voices. And so bringing new people to the field has always been an extremely important principle. And so if you're thinking about applying your skills to a disease-focused area, please do think about Safari. So, and of course, because I'm here, we have a particular interest in expanding our effort to systems, computational, and theoretical neuroscience. As I mentioned before, we've made incredible strides in genetics and cellular molecular science, and we really need to understand what the consequences are at the network level. And importantly, there's a huge plethora of experimental data out there on autism, but surprisingly, there aren't that many people that are taking that data and conceptualizing it within really rigorously defined theoretical frameworks and thinking about how we can use those frameworks to create predictive hypotheses and falsifiable hypotheses. So next. So like I said, since 2015 to date, we have 31 current fellows. They're doing extremely well. I'm happy to say that all of them have transitioned successfully to terrific positions. And the first ones are now just coming to the end of their award. So they're sort of in the past couple of years have been applying for R01s and other sort of coveted grants. And to date, the success rate has been extremely good, with many of them getting R01s and DP2s and other kinds of prestigious prizes. And lastly, I wanted to mention that in addition to the grant money, another really special part of the community is sort of the community building aspect of it. So what I like to say to my fellows when I have their welcome call is, your goal of succeeding in science is now my goal as well. And so we do a lot of different things in terms of giving feedback on job offers, having retreats, professional development workshops, and it's a really sort of rewarding community to be part of. So of course, we're here with the 2021 BTI RFA, and thanks to Laura for giving such a great intro to all sort of the ins and outs of how you can apply. Our policies are very similar to SCGB, but I did want to note two major differences in eligibility. One is that we do have a time cap on postdoctoral years. So you need to have 6 years or less of postdoctoral training, but this of course does not include any pauses that may be related to personal or professional leave periods. And given how COVID has impacted all of us, we're not counting the 2020 calendar year towards that cap as well. And just like SCGB, there is no citizenship or permanent resident requirement, but the faculty position does need to be at a US or Canadian institution. We may consider some exceptional circumstances, so please always feel free to contact us if you have some special case that is worthy of further discussion. And lastly, I just wanted to do a shout out that some of you may be interested in applying to both and want to know about that. So you can, we do allow you to do that, but we have really independent review processes. And so those applications will be independently reviewed. So lastly, I just wanted to remind everyone that the RFA is open now. Applications are due April 12th, and we'll announce our fellows in July of this July. And here's my name and email. Please feel free to contact me if you are interested in applying. Happy to take questions.
Thank you very much, Alice. We do have quite a few questions. The highest trending question is, how long will the Bridge to Independence Award be around for? Multiple cycles or only this 1 year?
For us, it's been around every year for the last 5 years, and it's a standing grant program. So if you're not applying this year, you can think about it in 2 or 3 years.
Laura? You're muted still, unfortunately.
Thank you. A classic mistake. Yeah, it is SCGB's first year launching the program. We have been discussing it as something that we are planning to start this year and continue going forward. But exactly what that trajectory will look like is still to be determined.
And is that policy that Alice just mentioned of not counting 2020 as a year, which I think is a perfectly reasonable policy, is that a Simons-wide policy, or is that just for Safari?
Well, the 6-year cap is just for the BTI program in general. So we don't have a postdoctoral cap, and then like SCGB doesn't have a cap at all. So it's really only, I think, pertinent to the BTI.
All right. How many of these awards will you give out typically in a given year? I think for both of the grant schemes, this is an interesting question. And what's the expected success rate? Laura, your video just cut out, just so you know.
Oh. Well, I can comment on Safari while Laura deals with technical glitches. So we're really lucky that we don't have to predefine slots. We can't say, oh, we only have room for 2, or we only have room for 5. So between 2015 and now, it's been somewhere between 3 and 7 fellows have been designated every year. And it's really a sort of robust discussion that we have with our external review panel on who we think really fits the program and is really a promising independent fellow. So in terms of rates, it's highly competitive. There's no doubt about it. So I would say somewhere between 10 and 15%. I
assume it's probably the same for the Global Brain Initiative.
We are currently participating, and I've just cut out, if my video isn't working, I'll just go with audio, but we are currently anticipating between 3 and 5 fellows in this first year. Also, just rolling back, the 2020 not counting against you certainly applies to our Bridge to Independence, but we do not have a cap on number of postdoctoral years. So it is something that we're certainly flexible with, both in terms of what we see on your application and time, but certainly SCGB considers that as well.
In the same vein, someone asked whether the 18 months requirement starts counting after the first submission deadline or after notification of the fellows on the 30th of June.
Notification on our part, but I will also notice that is, we encourage fellows once selected to communicate with us and staff. We certainly, our goal is to help fellows secure those positions and activate those awards, and so we can be flexible as needs allow.
Another question is regarding the profile of the fellow you're looking for. Is there any certain profile? Are there publications, project ideas, previous grant support? What makes a successful fellow?
So I can chime in just from experience. So the way we think about the review criteria are person, project, and relevance in that order. So person is probably what you're really talking about in terms of the profile. And yes, absolutely, publications, previous grant support, that kind of thing. And letters of recommendation is also a part of the application. And so thinking about whether or not somebody really is at the sort of pinnacle of their postdoctoral traineeship and are ready to make that next leap, that is really a defining characteristic, whether or not they've shown productivity and an ability to be both creative, but also grounded in their work.
SCGB has a very similar approach, and in particular, I would emphasize that we are really looking for science and projects that are aligned with our scientific mission. So thinking about understanding neural dynamics and coding, and especially doing so with methods that have high spatiotemporal resolution.
Thank you. Another question pertains collaborations, especially for theorists. It's known that often theory applications are not successful unless theorists will be able to point at 10 or 15 or maybe 20,000 different experimental collaborations that they can successfully participate in. Sorry to be polemic, I'm one of those theorists that needs to show lots of collaborations to get money. How important will you find the pointing out of potential and existing collaborations with experimentalists for theory candidates?
Testing if the video will come back on. Let me know if I need to turn it off again. SCGB has a lot of experience working with and funding theorists, and so we certainly encourage them to apply. And our team and our review committee are both very familiar with what that process looks like, including some theorists themselves. So we certainly encourage applicants to include known collaborators and those that they anticipate continuing to work with, but it is something that I think our committee is well suited to understand and recognize and encourage.
So for Safari, we're certainly newer to supporting theoretical science, theoretical neuroscience, which is part of why I'm here. But at the same time, we have, for example, extremely high quality genetic data from autism cohorts right now. In addition, we have very deep ties to the autism community. So in terms of thinking about career development and making connections with people, those are things that we do on a daily basis with our senior scientists already. So it'll naturally extend to junior scientists. I would just say that at the application point, of course, we want to see that you made a good faith effort to understand the autism literature and to be really thoughtful about sort of the issues that are in the field. But in terms of actually making the connections, that is less of an issue for us. And then having a deep history of collaborating with experimentalists than it is being a thoughtful sort of idea person.
Maybe in the same vein, is there any experimental technique that you categorically exclude?
So I don't think this is pertinent for this group, but this is mostly, we don't really focus necessarily on improvements in behavioral interventions or psychosocial interventions, although I don't think this is going to be hugely applicable to this community. So. But
so, for example, primate work or.
Oh, no. I meant like thinking about family factors, social stress, community stress, things like that. Absolutely. Across all species, we support work from C. elegans through to people. Absolutely.
We don't have any categorical exclude is a strong term, but I will note that SCGB has historically so far funded projects at single cell resolution. Though I also do want to highlight, in terms of thinking about the future of the program, we have an investigator pilot award open now, which is you can also find on our website, which talks about some of the possible avenues for future direction of SCGB funding, including things like connectomics, expanding into perhaps not single cell data, but again, it's still very high spatiotemporal resolution data and things of that sort. And in terms of model organisms, we already fund primate work and a number of different model systems. You can also see the 20 collaborative projects that we currently fund on the website for an idea of the scope and purview of where we've been and looking into the mission of that new pilot award for where we're going.
Laura, there is a follow-up question on that. Someone is asking, they're working on retina, so they're working on, peripheral nervous system. Would something like that be funded under the Simons scope?
It sounds like what I would say in terms of if, say a specific system, a specific question, it's really about whether you are looking into the types of things we want to understand about the brain and encoding, so neural dynamics, neural encoding. If that's what you're looking at in a retina system, I think that that's something we'd be interested in reading about for sure.
A few people are asking, including myself, whether you grant grants to non-US institutions and non-US fellows. So could you apply from another country other than the United States?
So this is a spot where there's a little bit of a difference between SCGB and Safari. Both programs welcome applicants from any country currently, but there is a key difference in terms of where the award can actually be activated. For SCGB, we don't have any country limits on where the tenure track position needs to be, but for Safari, that's currently in US and Canada. But both programs welcome applications from applicants independent of nationality.
Thank you. What happens if I'm not able to secure a fellowship or a follow-up position within 18 months? Do I have to pay the money back?
As I mentioned before, this is a space where we encourage fellows to be in touch with the staff as they're going through the job search process. We are hoping to get applicants that are looking to enter the job market, as opposed to someone who's a little bit earlier in the process and not planning to make that step for a few years. But we do want people who are looking to enter the job market, but then if we are very flexible and want to help you get that job. We want to provide the letter, but we also want to provide advice and support along the way. And so we are flexible with our fellows in terms of exactly how that process shakes out for them in terms of 18 months or a little longer. Alice?
Yeah, and just in terms of past history, this has certainly gotten, certainly had examples of the situation where people, a publication got held up, there's a two-body situation, there's lots of completely understandable reasons why somebody may not make it in that first round. And so particularly because of the job market now, we know that it's already anticipated from my end that a good portion of people will also be applying, possibly be trying again in the next cycle as well. I definitely work very closely with the fellows on reviewing offer letters and negotiating with the institutions. So far, I have not had anybody that has not successfully transitioned within 2 years, but it's always a possibility. The issue about returning the money is actually not so pertinent here because the money doesn't actually start flowing until you become an assistant professor.
Someone was asking if there's any special circumstance which would allow them to bring a Safari grant abroad from the US.
For our senior, so it kind of depends on exactly how that question is asked. For our other Safari awards, we have like, other kinds of grant mechanisms for established scientists, and those we fund globally. It's just the BTI is a little bit different, but there could be an example. I think I'm seeing here like an example circumstance is somebody who has a two-body situation. So please come talk to us. We don't want to ever, we know this is extremely important and complicated situation. So it's one of those situations where it's better to reach out to us than to assume.
Laura, I have two more questions for you. Number one is how much preliminary data is expected in a fellowship application? And then number two is whether the 4 points of the SCGB mission are roughly weighed equal in terms of the evaluation criteria, or if you pay more attention to one of them. And if so, which point? Sure.
In terms of the mission, I think that, again, looking across the scope of projects we currently have, not all of them are hitting every point. I wouldn't say that one is way outweighed of the other. It's this overall scope of how we're trying to achieve that greater understanding of internal states in the brain and coding and dynamics. So I wouldn't say that there's a heavier weight on one than the other. In terms of preliminary data, think of it as your research plan. So there are no hard limits or requirements on this, but we want to know that we want to get a sense of the work being feasible. So it's not a hard and fast rule, but whatever you can show us to help us understand the scope and idea of your research plan, and also the feasibility and where those plans are coming from.
In the same vein, maybe, how do you balance risk and reward? I mean, some grant agencies are asking for a lot of risk and reward, and others want more butter and bread, or bread and butter. How much do you want extraordinary vision, and also how much do you want people to stay in their lane, so to speak? So how much does the track record of the applicant play a role?
I think that, so this is our first year for review, so I can't speak in terms of what I've seen in this particular review process in the past, but if what I understand of the conversations in developing our particular Bridge to Independence, big inspired ideas are great, but making sure that we understand that is grounded in work that you have done and a knowledge base that you have already established. Not that you have to stay exactly in what you've always done, but that you have built the skills, and it's a logical next step of your program. And big inspired ideas are great as well. Was there another component of that I missed?
Preliminary data, expectation of preliminary data.
Oh, yes, sorry, I thought I mentioned that a moment ago. Preliminary data is something that is great when it's available. Again, we're really looking to know that the work is feasible, and if that's coming from your preliminary data, then that's great. But that can also be demonstrated through reference to work you've done with others or previous studies, things of that sort.
It's very similar for Safari. I think one of the things, it's always an interesting balance, is that you want things to be grounded, right, in terms of feasibility, in terms of being knowledgeable about your own field and the field that you are trying to apply your proposal to. But you also want to differentiate yourself from your advisor. You don't want to just sound like a staff scientist. So you want to demonstrate and put out your best ideas. What makes you excited? What's creative for you? But it also has to be something that's feasible in the sense that, do you have the requisite skills, or are you very close to learning them? And will you be able to pull off something in the next 5 years? It needs to be something that you can convince people of, and you can also get people excited about.
Great, thank you. Laura, Alice, I think we're almost at the end of our time. Shervin, you asked a question that was answered already. Is there a postdoc year cap on the global brain? No, there isn't. Only for Safari is there a postdoc cap. But otherwise, I think we've answered all the questions that were asked. And we're spot on at 7:30. So I think it's, unless you want to entertain us a little longer, we're good to now all proceed into the beer section of the evening, if only there were one. I have a beer here, but it's only for me. And with that, I think I will let you go. I would like to thank you for supporting Cosyne and also for supporting neuroscience more generally. I'd like to thank the organizers and the background team. So the viewers have not seen that there is quite a few people hectically, and maybe not so hectically because they're much more professional than I could dream of being, arranging questions and giving technical help. So thank you also to the backstage team. And then, of course, thank you to the speakers that so gloriously entertained and educated us for the last 2 hours. And with that, have fun at the rest of Cosyne. This was session 2. Peace and out.
Thank you.
Bye.